As George just said, I completed recently my PhD as part of ImageInLife and hosted in the company Acquifer. And since this year now, I'm working as a data scientist in the company. But today I will talk about some achievements of this PhD. So I was working on image analysis, image processing, basically to facilitate the visualization and analysis of large screening data sets, mostly for zebrafish screening projects. And so just to give you an idea of the type of data I was working with, so as you can see, you can recognize the zebrafish larvae, and here it's image on a wide field microscope, and we mount them in well plates, 96 well plates, and so you can recognize on the edge of the well, and the fish are mounted in a gel so that they have like a nicely aligned position in every well. On the left side, you have like a preview of such acquisition at higher resolution. So we either go for dorsal orientation, like at the bottom, or lateral orientation. And we also do multi-dimensional acquisition, so either multiple channels or time points. And as you can imagine, since we have 96 wells, and for each of them this amount of dimension, this yields a lot of data, and therefore you need some kind of software solution adapted for their visualization. So that was the focus of my project. So as I said, we're using this gel to orient the larvae, and they are nicely aligned, so at low resolution, this is no problem. So typically for a screening project, we will start with low magnification objective, so we see our full samples. But as you see, there is always a slight positional shift, which is below the range of the millimeter, simply because even though we can see the larvae at the naked eye, and when we're mounting them in the plate under the bino, they are still very tiny, and it's virtually impossible to be so precise in the positioning. So at low magnification, this slight positional shift is not much of a problem. But if we want to go for higher resolution, let's say I want to image the head region at higher magnification, the field of view is much reduced, and therefore, if we just use the same coordinates in every well, we don't manage to catch the organ of interest. So one solution to that is to use automated microscopy or feedback microscopy, which is basically starting with this low resolution imaging, and then having some detection algorithm to localize the region of interest and communicate back the position of this region to the microscope. So in this way, you can have either a fully automated workflow, where if you have a good detection algorithm, then the user really does not need to take some action in between the two acquisition between low and high resolution. Or you can have, let's say, a relatively quick quality control from the user after detection, which is already saving time compared to having the user tell the microscope where to go, basically for every well position. So the question there is what detection algorithm to use. So there are a lot of them around. So I was looking in the literature and also playing a bit with what's available, and I came across template matching, which I really like because it's relatively simple and was already giving acceptable results. So the way it works is that you provide a template image of the object you're looking for. It's this small square image at the top left. And here we are looking at another type of sample. It's Medaka embryos, another fish model. And since they are circular, they tend to roll around in the well. And of course, they have random orientation and position from one well to the next. So therefore, it's interesting also to localize them. And so with template matching, the way it works, as I said, you give this template image, and the algorithm will scan basically the image in this kind of zigzag approach. And at every position, compare the pixel intensity. And from there, you get this kind of heat map, which tells you where there was a nice correlation between the gray values. And as you can see, so for the template, I was actually using the middle sample, the middle specimen. And you indeed see like there's some hotspot there. And from this correlation map, we can do some pre-post processing and isolate the most probable position. But as you can see, with a single template, we only had like half satisfying detection, and the second half were kind of shifted. And that's kind of a built-in limitation of template matching. It's not rotation invariant or other kind of change basically in your sample will tend to decrease the score. Although it's a relatively simple method. And for case where there is no rotation, for instance, it would still work quite nicely. But then I was thinking of a simple way to improve this method, to improve its robustness. And the solution, so the method we came up with, we call multi-template matching, or MTM for short, is instead of using a single template image, we can search with multiple template image, for instance, rotation of the original image. And therefore, we can increase the range of patterns that are detectable, and in turn, potentially improve the detection. And as you can see here, I just rotated my initial template 4 times by 90 degrees every time, and this was already enough to localize the embryos correctly. And well, the only difference between before with the single template matching is that this time we have multiple correlation map instead of a single one, but the processing them is the same. We just merge all this detection together. So basically, we managed to improve the robustness of the method at a low cost, let's say, and still being quite simple to understand. It's a bit like doing data augmentation with machine learning. And everyone, yeah, it's very easy to do this kind of augmentation, either rotation and flipping. Already, in most cases, would be improving the detection. So this is published, and there are also YouTube tutorials available, and the solution is available in Fiji as plugins, as a Python package, and also as a NIME workflow relying on this Python package. And the source code are also available on GitHub, so it's open source. You can have a look at it and even contribute it if you want, or extend it. So we then tested this on our data set. So with actually the good old template matching, as I was calling it, with a single template for the head region, it was already working quite well, even with slight change of morphology or grayscale variation in the image, illumination changes. And then with our Medaka embryos, this time doing flipping and rotation of the template, it was also working quite nicely. So this is the montage of the result for 10 different images, each of them containing 4 embryos. And ultimately, you can even use it for cell detection. Even though I tend to think intensity-based method like segmentation would work better. Is essentially because usually for cells you want to have the outline of the cells, and with template matching you always get a rectangular region of interest. Okay, so we went further and we implemented the method in our software at Acquifer, in our so-called plate viewer. And we did this kind of semi-automated microscopy that I was mentioning initially. So this is the view of the overview experiment when we see the full well. And from there, the user can define his region of interest. In this case, it was his kidney region, again, labeled with fluorescence, and can run the detection then within the software and directly get the detected region. And from there, start the high resolution acquisition, which looks like this. So this is all very nice. And so multi-template matching was one of the outcome of my PhD. And then I was thinking, I was investigating other object detection methods. And actually first came across a limitation, let's say, in existing software. Basically, I needed to annotate different object categories, let's say different tissues or different categories of cells, cell population. Let's say here in blue, you have one population, and in pink, you have another population. So you could do this kind of color coding previously, but it was not very user-friendly. And so I thought, why not associate some category to each region of interest? And so, as you know, ImageJ and Fiji are open source projects, so you can contribute to the source code and new functionalities. And so that's what I did. And it's now available in ImageJ and Fiji. So you can basically, if you update ImageJ, if you go to this menu, you can define the current category, which is basically represented as a number, this group number, and then annotate your object with this group. And so this group, the group is this number that, as I was saying, and it's associated to a color, which is predefined. You cannot change it. So that basically all objects from the same category have the same color, quite user-friendly. And you can also define a name. So I'm not going too much into details. If you want more information about this, you can go on the F1000 research website. In the New Bias Gateway, there are some slides about it. And also on the Fiji YouTube channel, I recorded some tutorials. But basically, what you have to understand is that we can now easily annotate different categories of objects within ImageJ Fiji. So this was actually quite some progress, but then I thought I could maybe push it further because I wanted to annotate basically a region of interest with very similar sizes. You know, like these Medaka embryos, for instance, they all have pretty much the same radius. So I thought, why not having like some kind of template region of interest? It would be also interesting when we are annotating bands in electrophoresis gels or dot blood. So I did this small tool set, which I call the RI one-click tools, which is also available for Fiji via an update site. And basically you can, as I was saying, define the dimension of your RI in advance and optional action that you want to perform automatically upon clicking, like adding the RI to the RI manager, running some measurement, or going to the next slice. So this is also published. You can have a look at this micro publication, but I think the video will tell you, will showcase a bit more in a more intuitive way. So here I'm defining my dimension for my region of interest. It's a circle of 15 radius pixels. And as you can see now, I can very quickly annotate my dot plot. And for every click, this is creating the ROI and adding it to the ROI manager and running the measurement at the bottom. And as you can see, the area is the same, obviously, because they are all the same shape. All right. So this was great to do annotation of categories with region of interest or speed up annotation of region of interest for, let's say, object detection, this kind of stuff. But I also needed to do image classification without region of interest. So just saying this image is part of this category, or describing like this image, it's representing this or this phenotype. So also came with another set of tools, third one, let's say. And so basically there you have plugins for which you can define your categories in advance. So in this case, I was just annotating the mitosis stage. And you can add then your stack, click the button, and this is adding automatically to the result table with the selected category. So this plugin is doing, for instance, a single category per image or per image plane if you have a stack or hyper stack. But I also have a version where you can have multiple categories. Again, you define them in advance. And as you can see now, for every of these two images, we can report multiple phenotypes and for each of them differently. And from there, you can do very complex annotation, either just for classical, sample exploration, or if you want to do like a training of an image classification algorithm, you can do your ground truth annotations. And we actually showed in the paper, because this is also published, that we could train then a deep learning network to recognize these morphologies. So if I zoom a bit, you see they're slightly different. This one is a bit more blown up, and this one, again, these are kidneys, is a bit more narrow. But if you're not an expert, like it's maybe not super obvious, but we managed to have an algorithm do this classification automatically. And finally, right now, so as I was saying, I'm working as a data scientist, and I'm basically developing a custom plugin in Fiji for the visualization of our data set. And so now we are having a bunch of plugins for that, for batch processing, visualization, and so on. And this one is like for visualization of full plates. So multiple well position and multiple dimensions. So if you have a time lapse, multiple channels. And it's using this virtual stack technology, maybe, which is available in ImageJ, maybe you heard about it. But basically, this montage image is not existing on disk. It's generated on the fly from the data. So each of these images is like 2000 by 2000 pixels. And then you have to take into account multiple channels at time points. But with this virtual stack, we generate this image on the fly in memory. So it's really quick. And so you can, even on a small laptop, you can visualize your data very quickly in a user-friendly way. And this is really helping our customer, but it's really then tailor-made towards our data set. So if you want more information about that, you can always check our website. And with this, I'm finishing my presentation, thanking my past and current colleagues, of course, Joran, my supervisor, also the contributors to the open source software who are making a great job at maintaining them and always improving the functionality, the funding, and as well the people of the consortium of ImageInLife for all this administrative work and this great experience. And thank you for your attention. I'm happy to take any question.